Hi, I'm Arup Acharji from Proteomics Lab, IIT Bombay. Welcome to the summer camp. Today we shall learn about reverse transcription polymerase chain reaction, popularly called as RT-PCR. Before we go on to the technique of PCR, which you are well aware of by now, let us try to understand the central dogma of molecular biology. The central dogma of molecular biology states that genetic information usually is stored in the form of DNA. This DNA replicates, and the DNA replication will produce more of that DNA. Thereafter, we have a process called transcription. What does transcription do? It converts DNA to RNA. Whereas, after that, we have another biological process called translation, which converts RNA into protein. This was a well-established fact. But very soon, scientists discovered the phenomenon called reverse transcription. Here, a specific enzyme is used that converts RNA to DNA. Reverse transcriptase PCR basically hinges upon this very fact that a few enzymes can make DNA once it is given the RNA template. So let's look at what reverse transcriptase PCR looks like. So consider this as the RNA. RNA is a single-stranded molecule. When it is heated to around 70 degrees Celsius, primers are added. Once the primers are added, we have an enzyme called reverse transcriptase. This reverse transcriptase enzyme will use this RNA as a template and produce DNA. This DNA is called cDNA or complementary DNA. After a few initial rounds of RNA to cDNA, you can add a normal polymerase like Taq polymerase, and then Taq polymerase will allow for the amplification of the DNA. Now, once this is applied in conjunction with another technology that we would be talking briefly about, then we can observe the concentration growth of amplified DNA in a real time. So how does real-time PCR work? So think of the DNA, and this is the reporter probe. Now, if you carefully look at the probe, there is a quencher sequence, then there is a reporter sequence. When the primer binds with the DNA, there is the extension step. Extension is usually done by the Taq polymerase that uses DNA as the template. When Taq polymerase comes to the region where the probe binds, what happens is, due to hydrolysis, this reporter is dislodged. So the quencher and the reporter, which were earlier very close to each other, moves apart. And as the quencher is also cleaved, both quencher and reporter are now in a free solution state. So they are not they are not very spatially close. So the spatial distance between the reporter and the quencher molecule goes away. As soon as this happens, the reporter starts emitting a fluorescent signal. So why do I discuss this with you? Let us see how RT-PCR as a technology has been applied to fight COVID-19. You must be aware that many of the government institutions have made RT-PCR reports compulsory for travel. You need to be RT-PCR negative. So what is this RT-PCR all about? How do they do it? For COVID-19 testing, usually the sample for the virus is taken by a nasopharyngeal swab, because it is in this upper respiratory tract that the virus usually stays during the initial part of infection. That is transferred into a solution called VTM or viral transport media. RNA is extracted from this viral transport media using automated RNA extractors. Thereafter, using a qPCR machine as described here, like QuantStudio 12, we do quantitative PCR. And once quantitative PCR is done, we see whether the sample is E gene or ORF gene that are specific to SARS-CoV-2, if they are positive or negative. Now let us look at a live demonstration of how real-time PCR for COVID testing happens. So this video shows you that how these VTMs are arranged inside a BSL-2 laminar flow. What we see here is VTM been poured into these small chambers. These are 96 fill chambers where we have already put a buffer called as the viral lysis buffer. Now we are adding the viral transport media. This viral transport media is very essential because it is containing, or might be it is not containing, the virus, whereas the lysis buffer inside will help the VTM, or the viral nucleic acid, to come into solution. The next step is to put them in this automated RNA extractors. So you see there are multiple plates lined up. Now once the RNA is extracted, we add the PCR master mix that contains all the probes and enzymes required. And thereafter, this instrument will do the needful. Once the PCR cycle starts, we wait for the instrument to give an output directly on the computer. So you see the output is now available on the computer. Let us see how the output looks. Yes, so the output looks like this. You see there are genes like E gene, ORF1 and RPP30. Now RPP30 is an endogenous control. So this is a gene that is expressed by humans. So if the sampling was proper and the RNA extraction was proper, this RPP should always be positive. This means your entire protocol that you have done, right from sampling the patient to the RNA extraction procedure, everything has worked properly. Next is E gene and the ORF1 gene. ORF1 is indicated by the red line and E gene is indicated by the blue line. These sigmoidal curves are basically the growth curves, how the DNA got amplified. So from the viral RNA, we are converting it into complementary DNA, and then that complementary DNA is using 3 sets of primers against E gene, ORF1 gene and the RPP30 gene. And we have 3 different probes. These 3 different primers, while extending their amplifying the respective targets, give out the fluorescence by hydrolyzing 3 different probes. Now you have 3 different growth curves. So RPP30 is present here, which means the sample was extracted properly. Whereas we also see ORF1 and E gene curves are also positive, which basically means that this sample is from a COVID-19 positive patient. Hope you found this small tutorial interesting. Thank you.